Welcome to All the Best, an interview series with UNL Orca awardees, where UNL means University of Nebraska-Lincoln, and Orca is the Outstanding Research and Creativities Award, which is UNL's top award. In Gerda Lerner's book, Why History Matters, she explains that life stories give meaning to the present and the future because we identify what is important to us. And she says, our stories show how we are interconnected in a particular time and place. As for this interview series, Orca awardees have experiences that reach back more than a half a century, which will give us a personal view of how the best research emerged from UNL. It is my pleasure to introduce today's interviewee, Emeritus Professor Pil-Soon Song. He was awarded the Orca in 2000. After I provide a quick summary of his life and career, Pil will tell us about his time at UNL, starting with his award-winning research. Pil was born in Osaka, Japan. When he was 13, his family moved back to Korea. Something must have sparked an interest in science because he enrolled at Seoul National University, where he earned a bachelor's and master's in agricultural chemistry with an emphasis on biochemistry. Next, he earned a doctorate from the University of California at Davis and did postdoctoral work at Iowa State University in Ames with David Metzler. In 1987, after about 21 years as a faculty member in the chemistry department at Texas Tech University, he joined UNL Chemistry as chair and full professor. He is fondly remembered because he led us through a decade of faculty hiring, including myself in 1990. There are two interesting facts I'd like to mention before we get started. One is that he was a leader on many levels in the international photochemistry and photobiology community. The other is that he published over 200 journal articles and co-authored over 20 patents. He left in 2003 to develop his turfgrass project at Jeju National University in Korea. With that, I want to welcome Pil to the interview. So welcome, Pil. Let's jump into the first question.
Okay.
What were your impressions about the UNL campus when you saw it for the very first time and when you saw it last?
When I saw it for the first time, I was impressed by the tallest building for the Department of Chemistry at UNL. When I was visiting for the chairmanship of the department, yeah. I think that was in 1987.
Okay. And then let's jump right into your award-winning research. Give us a
Yes.
sense of how that came about, what you actually did or what you think you got the award for, and why it's important.
I was always interested in biophysical aspect of biochemistry, and in studying biochemistry, biophysics, I became interested in the relationship between life and light, and that led me to develop a research program for a long time, dealing with light and life, and which became what was later called photobiology. So that has been my longest lasting research program for my graduate students and postdocs and for myself. Yeah. And what was the other part of the question?
Well, so yeah, so tell us about some of the molecules that you analyzed.
Oh, most of at first it was about the flavins, including riboflavin, FAD, FMN, and FAD, and my interest in flavins can be attributed to the controversy at the time as to what the potential candidate is for the photoreceptor in phototropism in plants. And so my candidates, the candidates, people in the field were thinking about were carotenoids versus flavins. And I began to examine those compounds as light receptor, photoreceptor, and came to the conclusion that flavins are the most likely rather than carotenoids. And that's how it came about. And in later studies, when I say we, I mean graduate students and postdocs and myself became interested in phytochrome because that phytochrome is the photoreceptor modulating the plant's response to light. And in the program that I was pursuing with my postdocs and graduate students at the time was phytochrome mutant, which artificially generated using genetic DNA substitution. And looking at the various combinations of substitutions, we found one particular one that was most interesting to us at the time, and that's phytochrome, oat phytochrome, serine substituted to alanine. And that mutant, 598 alanine mutant, behaved like a behaved as if it functioned in the shade. And so it was dark activated mutant. And so that's the motivation to look into its effect on mowing of the lawn. Because in the mowing of lawn, the neighbor shading produces the dark environment for the plants. And phytochrome mutant that we generated was acting under those conditions. Yeah.
Yeah, so interesting. So when I was in the department, you worked on stentorin and other, what I think would be
Yes.
unusual photoreceptors. And so what was that all
Yes.
about?
Yeah. We worked on photoreceptors for photo movement in amoeba, including stentor and blepharisma. And so the structures of the photoreceptors for this unicellular organism were not known at the time. So that's how we decided to look at the structures of the photoreceptors, and we found them and published the results.
And so a lot of your work involved lasers, is that right? And then you look at the excitation and decay of the light absorption?
The later studies included the picosecond spectroscopy, which I learned and studied at the Molecular Institute of Institute for Molecular Science at the Okazaki National Laboratory in Japan
Oh.
when I was on sabbatical. So I spent
Mm.
my time with the institute at the time.
Mm.
So the spectroscopic, picosecond spectroscopy, and circular dichroism
Mm.
were
Mm.
those two special instruments
Mm.
and that we were using. Yeah.
And so when you say photoreceptors, you're not really talking about the photosynthesis. You're talking about how does the plant know where the sun is, right?
Exactly. Yeah. Yeah. Because stento avoids light.
Ah, yeah.
And in order to make that movement away from light or
Yeah.
shaded area of incubating medium,
Mm.
stento and breviarisma
Mm.
use their photoreceptors.
Mm.
And those photoreceptors are, I don't know if you know about hypericin.
Yes. Yes.
Hypericin.
Yeah.
That's a natural photosensitizer that
Mm.
people can get photosensitized and develop some psoriasis
Oh,
on their
wow.
skins.
Oh.
And so the photoreceptors of these two organisms used stentorin and breviarismin, which are chemical derivatives of hypericin.
Oh, I get it.
So the amoeba cells were avoiding the light by using the same kind of uh, photosensitizer.
And so both of those molecules, are they high quantum yield, or I mean, what makes them special? Or is it just chance that those particular molecules were used?
Uh, I guess when just that question of evolution, when
Yeah.
these unicellular organisms were evolving, they must have figured out how to survive.
Yeah.
And in order to do that, they had to avoid this very strong natural photosensitizer,
Mhm.
hypericin.
Whereas all organisms have riboflavin and cytochrome P450s.
Yes,
Yeah.
but but they don't necessarily they are not necessarily photoreceptors for these unicellular organisms. Yeah, uh,
Mm.
they may be photoreceptors in plants phototropism, uh, which is moving toward light,
Mm.
positive phototropism, and moving away from light, that's negative phototropism. So some plants have positive, some plants have negative
Mm.
phototropism,
Mm.
and flavins are probably very important there.
Mm. Okay. Well, tell us about one or two of your graduate students, postdocs, or visiting professors. What were their
Okay.
projects that you remember and what happened in
Uh,
their careers?
my very first graduate student was Tom Moore.
Okay.
Uh, he was an undergraduate and a student enrolled in my first course that I taught at Texas Tech.
Wow.
That's biochemistry, and he was a math major, so he registered for the biochemistry course for some reason, and we and he became interested in biochemistry,
Very
and
brave.
later became very famous. In fact, his research articles were featured in Science a couple of times,
Wow.
and Nature also.
Wow.
And now he's a distinguished endowed professor at Arizona State University.
Mm.
He might have retired by now.
Yeah, right.
And the other student is Dave Farrens.
Oh, okay.
You, how did you know about him?
Yeah, I met Dave, right? Wasn't he at
Yes,
UNL?
Yeah, that's right. Dave Farnes, became eventually professor of biochemistry at Oregon Health Science University.
Okay.
And third one, let's see, Bill Parker,
Mhm.
who who is now a professor at Duke University Medical School.
Mhm.
And exactly what he is doing now, I don't know. He's a completely different field of science and medicine.
Yeah.
So that's Bill Parker. And I guess those are three uh, most distinguished
Mhm.
graduate students from my group.
Yep.
And there are many uh, group, uh, many research participants from Korea
Oh.
who later, or most of them came back to Korea after their graduation or
Mhm.
completion of their postdoc experience in the United States. And
Mm.
some of them have done quite well in Korea. So
Mm.
I yeah, that's it as far as my graduate students and postdocs are concerned.
That sounds good.
Yeah.
So, what do you remember as your most unexpected research finding, and did it take your lab in a new direction?
Yeah, as I was mentioning earlier, that would be uh, old phytochrome uh, 598
Mm.
mutant.
Mm. Mm.
And because of its activity in the shade, I became interested in its potential technological applications. Uh, and if that's successful, what it means is that was that the lawn in your front and backyard need not be mowed so often
Yeah.
because the plants were responding to this mutant modulated by the mutant phytochrome.
Mm.
Yeah. And so that became so that led me to do more additional research, uh, continued at Jeju National University.
Okay.
And so I spent about 10 years at Jeju National University developing this particular mutant as the basis for biotechnological applications.
Mm. Mm.
And we applied mutant to zoysia grass, which is the common grass particularly popular
Mm.
for golf uh, in Jeju Island.
Yeah.
Yeah.
No, really exciting. So let's talk about your most cited papers. I just mentioned to you the two that I found. One was
Yes.
titled Phytochrome Signaling is Mediated Through Nucleoside Diphosphate Kinase 2. That was from
Yes.
1999, published in
Yes.
Nature.
Yes.
Has 383 citations. Now, I just want to interject here. In my opinion, if anything has more than 50, it's amazing. So this
Uh,
is way amazing. And you've got a lot of these high impact ones. Then also molecular luminescence studies of flavins, one,
yes.
the excited states of flavins. That's 1972. So tell us about the first one,
So on
phytochrome signaling
mitochondrial signaling,
through
let's
nucleoside
see.
diphosphate kinase 2.
Yeah. Uh, that particular uh, uh, compound, uh, uh, I I did not I did not pursue it further as
Oh.
far as its role
Mm.
uh, uh, is concerned. So I haven't followed even those people who use the
I see.
result in their own research.
So, but
And one
why,
reason
why,
for
yeah,
that,
okay. Why do you think it's
uh,
so cited though? What is so
uh,
exciting about it?
because up until that time, nobody had any idea about what the possible uh, photoreceptor could be for the plant's response to light. And the reason that I did not pursue it, in spite of many people using the result for their applications, it was that uh, Kilchu Choi was my graduate student, and he did most of the work, and he continued to pursue that line of research after he came back to Korea.
I see.
So I left that to him, and he has developed the research in that along that line. But later on, he expanded his own research beyond that. Yeah.
So did he come to UNL to do the work?
You mean
where was the work
student?
done? Yeah, he was a graduate student at UNL.
Yes.
Okay.
Yes.
And then how about the other one? Molecular luminescence on flavins.
Molecular luminescence of flavin did not. That was not a specific research program, but it's more of various aspects of flavins in terms of its photochemical reactivity and the spectroscopic properties as a photoreceptor. So there was one review article that I published. I don't remember exactly what year. That's probably the reason why it's cited very often. Yeah.
I see that Tom Moore is the second author.
Okay.
So we already talked about him. That's good.
Okay.
Then, do you recall any papers that you thought were really good, but that did not raise a lot of interest among the people in the community?
Yeah. Namely, structures of unicellular amoeba photoreceptors.
Oh, really?
In brephoresin and stentorin.
Oh.
These compounds, their structures we elucidated, but the structure themselves are not really cited by many researchers. That's probably because not too many people are interested in unicellular amoeba.
Sure.
Yeah, so they're not very much cited, in spite of my own feeling of being pretty good for having identified and elucidated the structures. Yeah.
No. Great answer. So the next one is a really big one, and this is how do university professors interact with everybody else. So when I go into editorships, patent applications, companies, books, and we'll start with editorships. Did you do much editing for journals?
Yes, I was editor-in-chief. I was associate editor of the journal called Photochemistry and Photobiology, an official journal of the American Society for Photobiology.
Okay.
And my associate editorship was succeeded by my own succession. So I became editor-in-chief for 10 years after that. Yeah.
Wow. So how many total years?
10 years. The editorship?
Yeah.
13 years. Yeah.
13 years.
Yeah. Associate editor and the editor-in-chief. Yeah.
Okay. And then what is involved in editing? I mean, why do professors do that? And again, the podcast is for people who aren't professors.
Yeah, I guess it's kind of a way of expressing your gratitude to the field in which you were recognized in various functions. And so it's kind of a expression of gratitude to my fellow photobiologists. Yeah.
Oh, great. So patent applications, they are really complicated. I found in the US patent system, you've got about 23, but
Mm.
that your very first one was titled monoclonal antibodies for metallic cations, and it was from UNL. Fred Wagner was the lead, but there are a number of other professors on here. Tell me about that grant. How did that, or that
That patent,
patent?
I had no no significant role in it.
Okay.
So
But how did it influence how you did the next patent?
no, because that was not really uh, my own field
Yeah.
of interest at the time, and I guess my name was included in there because I was a member of that group at UNL at the time.
Mhm.
It's an inter-campus group made up of uh, people from what we call the East Campus because of its location,
Mhm.
and the City Campus, which is the main campus in Lincoln.
Mhm.
So
So you've got 23 patents. What are the other ones about?
those patents were, the first patent was on monoclonal antibody, and subsequent patents were all due to the directorship of the Kumo Asiana Institute for Environmental Science. And I was director, and as a result, virtually all of the patents coming out of that institute uh, listed me as the principal investigator. So
Uh,
many of those patents, except those involving lawn grass, uh, uh, were done by uh, the within the directorship. There is various groups, and one of the groups is the photobiology group, which included, as I mentioned earlier, Kilsu Choi was one of them. Yeah, he's the one who discovered NDPK.
yeah, 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 yeah.
Yeah.
So how about a company? Did you start any companies?
No, I
No.
didn't start any company.
Yeah. Did
I
you? Oh,
served as a director of the company institute for about 10 years.
oh, okay. How about books? Did you write any books? Did you edit any books?
I don't actually, it's more of a joint authorship with Chen, Mr. Dr. Chen. He graduated and became postdoc, and later he moved to Florida, and he and I assembled what's called photoreactivity indices, uh, in which each biological molecule was analyzed by quantum chemical molecular orbital method, identifying reactivity at each position of the biomolecule.
Hmm.
So we published this book as a monograph type book
Hmm.
in photoreactivity of not photoreactivity, bio reactivity of uh, reactivity indices of biomolecules. That was the title of the book. Yeah.
Hmm. Now
And
that
it was
sounds.
published by Texas Tech University
Oh,
Press.
okay. So let's talk about funding. Which organizations funded your research and how did that change over time?
uh, my research was funded by uh, NIH, NSF, what was the name of that funding administered by private research organization? What is that?
Nebraska Research Institute, maybe?
No, there was a name for it. It was a Career Development Award type of award was offered by that private foundation.
Okay.
I forgot the name of the foundation.
Yeah.
Yeah.
And what techniques and instruments did you use in your research? We talked about lasers
Yeah.
and circular dichroism. What else?
Instruments?
Yeah.
Yeah.
And
I mentioned
techniques.
earlier. Yeah. I mentioned earlier picosecond spectroscopic technique. And that I didn't have my own instrument in my laboratory at UNL, but I did it as on my sabbatical at the Institute for Molecular Science in Okazaki, Japan. And that continued as long as I had interest in photoreceptors, including phytochrome and flavins and stentorin and so forth.
Yeah.
So I continued that research at Okazaki for about more than 10 years, I think. Yeah.
Oh.
And my host was he eventually retired from the institute, and I also collaborated with Professor Masaki Furuya, who was at the University of Tokyo with the affiliation with Hitachi Institute in Okazaki, Japan. Yeah.
So really special instruments.
Yes.
And
circular dichroism was another one.
Yeah. Your students did a lot of isolation of the molecules, I believe.
Phytochrome.
Yeah, phytochrome, yeah.
Phytochrome. There were only 2 laboratories in the field. Nobody could isolate phytochrome effectively until people detected phytochrome and essentially in vivo with the plant tissues, but actually isolating it in pure form, we were the one of the two capable of doing that at the time.
Oh,
Dave
wow.
Farrens was involved in that. Jung-il Kim, who is a professor at Jeju University here in Korea. He developed this isolation purification procedure and produced something like 20 to 30 milligram per try. And the only other laboratory in the field was from Mülheim in Germany. And they were doing same isolation, probably not identical procedure, but slightly perhaps different from ours. And they were doing the isolation. I don't know how much, what kind of yield they were getting.
Right.
But anyway, those were the 2 laboratories, only those 2 laboratories able to produce phytochromes in pure form.
And so what made it so hard? Was it a membrane protein or just in low
No,
solubility?
it's a soluble protein.
Yeah.
Very, very subjective, sensitive to proteolytic digestion.
Oh, yeah.
So when you are, for example, blending your tissues,
Mhm.
that releases the uh, proteolytic enzymes,
Yeah.
and they attack phytochrome, including
Mm.
phytochrome, because phytochrome is a protein.
Hmm.
So that made it very difficult to.
Mm.
So one has to do two things. You have to isolate phytochrome in dark room
Mm.
with a little bit of green safe light and a cold room.
Mm.
So combination of cold room
Mm.
and very dim green safe light, the two conditions under which you isolate phytochrome in pure
Wow.
form.
Wow. Heroic. In
Yeah.
some ways, the next question says science is cooperative and competitive. So I'd like to know whether your work was ever scooped or did someone produce similar results at the same time?
Mm. No, not that I can recall. No,
Okay,
nothing that's outstanding to call it scooped.
sure.
Yeah, yeah.
So were your results ever contradicted, even by yourself? And then how was it resolved?
Uh, Contradicted. Yes, I think there were some errors in the uh, in different kinds of experiments in research, and those errors were, if they were minor, just I didn't do anything.
Mhm.
But if they were significant, uh, I just simply stopped using the same kind of method description section of
Mm,
research papers.
mm,
I didn't advertise this is my error. I
sure,
mean, it's a significant. It's not that significant part of the method description.
sure. So you never published a correction or anything?
No.
Okay. So we've described the essence of what you did, how molecules absorb light, why that's important to the organism. But is there anything else you'd like to add before we move on to other career activities?
Nothing really.
So,
Only uh, uh, regret, if I have, is the fact that the structures of photoreceptors in the unicellular amoeba
mhm.
uh, uh, have not been really um, uh, recognized by people in the field of photobiology. Yeah, yeah. So,
Yep, yep. That's just really an interesting observation. So, on the other career activities, we're going to talk about first societies. So, I know you were heavily involved in the one society, and I'm sure you went to a lot of conferences. Which ones did you go to regularly? And is there one that was especially memorable?
conferences.
Yeah.
Well, annually I attended, of course, American Society for Photobiology meetings,
Yeah.
and where as an editor, associate editor, and editor, I also conducted editorial conference at the time,
Mm.
along with the uh, Photobiology Society meeting.
Mhm.
Uh, let's see. I guess that's it. Yeah,
That's your main one, yeah.
yeah, main
And
one.
so where were those held? Were they always in the US or were they everywhere?
Most of the time in the U.S., in different locations of U.S.
Mhm.
I don't remember if we had any ASP, the American Society for Photobiology.
Sure, American, yeah,
Meeting was held outside of U.S.
sure.
I don't remember. I don't think so. Mostly in different cities of the United States.
Okay. So how about sabbaticals? You mentioned one sabbatical. Did you do others? And then it sounds like the one you described was very important for your research.
That's right. That continues for a long time.
Yeah.
Other sabbatical, I don't. No, I haven't done any additional sabbatical year. I
Okay.
mean, I had short visit to various laboratories,
Mhm.
but I cannot call them sabbatical.
Right.
Uh,
So service contributions. Um, you became the chair of UNL chemistry. So that's a
yes.
major service contribution, but I believe you were also chair for a while at Texas Tech, if I remember correctly. But what caused you to do that?
Well, after my for after a long um, uh, several years, about 10, almost 20 years. Yeah,
Mhm.
I spent at Texas Tech. They invited me as, by they, I mean UNL Department of Chemistry recruiting committee
Oh.
invited me to be chair of the
Mm.
department. So I suggested to them that I am interested in moving to other university from Texas Tech, but
Mm.
I wasn't interested in chairmanship.
Oh.
And so then, so I sort of declined. But then Michael Gross was the recruiting committee chair.
Mm.
He called me a couple of times and strongly advised me and suggested that I visit again. So on my second visit, I accepted the position.
Oh. And so service contributions to the university as a chair, what did you do for the university?
I guess my contribution to the university is through the Department of Chemistry.
Okay.
Department of Chemistry was a modest size department,
Yeah.
and fortunately, the dean of the college was very supportive. Incidentally, he was also very persuasive uh, in my coming to
Mm.
UNL as a department chair.
Mm.
He was very supportive of chemistry department. In fact, the dean was himself physical chemist,
Oh.
and the combination of that allowed me to recruit additional faculty members for the department. So it was about something like 19 or 20 members when I joined the department
Mm.
in 1987. But when I left, it was one time it became a rather strong department of 23 faculty members in
Yeah. Yeah.
the Department of Chemistry.
Yeah.
But when I left in 2003, they lost some. Well, actually, I lost some faculty positions by another dean
Another dean, yeah.
who anthropologist, and
Mm.
he didn't like chemistry, I guess,
Yeah.
and he didn't allow me to replace one of the retiring chemistry department faculty member
Mm.
with another faculty member in chemistry,
Mm.
and he didn't approve,
Yeah.
and that his attitude, the anthropology faculty dean, continued for a couple more years under my chairmanship. So I left.
Oh, yeah. No, I agree. So, but it sounds like it was not agreed upon ahead of time that you would be able to hire so many faculty. Is that right? If you became chair?
No.
Oh,
Yeah, as I said earlier, Dean was very supportive of the chemistry department.
yeah,
Yeah, he was being chemist. He was a chemist himself. So
yeah.
yeah, I took
Okay,
advantage of that.
good.
Yeah,
Which courses did you teach? I know as chair you probably didn't teach much, but what did you teach more often than others? And then what are your thoughts about teaching courses?
I I taught biochemistry every year,
Oh, you did.
and that was my first full-time teaching obligation.
Mm,
And when I was doing it, I enjoyed it.
mm,
Uh, One thing that I learned as an undergraduate biochemistry uh, professor, uh, my native tongue,
mm.
Korean, was kind of a hindrance uh, to the students,
Sure.
and and so I became aware of that, and later on I tried to uh, avoid that native tongue
Mm,
syndrome
mm,
in my course teaching.
mm, mm.
Yeah.
All right, let us move on to your youth. Where did you grow up and what was your childhood like?
I grew up in Osaka, Japan. I don't remember much about my youth food.
Yeah.
Uh, very um, obscure youth life. I had maybe one or two experiences that I constantly uh, think about.
Hmm.
And one example was that I used to go to my neighbor, and in the neighbor, in the cloth hanger, there was a futon
Oh, yeah.
uh, for drying,
Hmm.
and I uh, accidentally caused it to drop on the ground,
Mm.
and I got so surprised and shocked that I didn't know what to say
Yeah.
to the owner of the neighbor.
And
Otherwise,
yeah.
uh, I remember coming back to Korea when I was 13 years old, and it was kind of a very difficult time. For one thing, the food was very uh, scarce,
Mm.
not much to eat in Jeju Island at the time.
Mm.
So the food was very poor,
Mm.
both in quantity and quality.
Mm.
And so our family lived in this small city in Jeju Island, and later moved to main city of Jeju City,
Mm.
where I started going to elementary school and high school.
Mm.
And that's gradually Korea was recovering its very poor economic country to a modestly developing country. And so that was okay for
Mm.
my childhood.
Mm. So
Uh,
why were your parents in Japan and why did they move back to Korea?
they the life in Jeju Island at the time was very tough, very difficult time, and so they my father decided to find a job in Japan.
Mm.
In fact, many Jeju citizens immigrated to Japan at that time.
Mm.
So we arrived in Osaka, Korean community section of the Osaka.
Mm.
So there were many fellow Jeju
Mm.
the immigrants as well.
Mm.
So that's where I spend most of my time.
And this was the 1950s then.
That's right.
Yeah.
In 1936, I was born in 1936.
Mm. Mm. All right. Did you have a special interest in science or math when you were a child, or what sparked your interest?
I wasn't particularly interested in basic science or technology at the time, engineering at the time, but only field that I wanted to get into when I grew up was medicine.
Mm.
And however, when I expressed that wishes, my parents said no,
Mm.
because sending a child to medical school at the time was extremely difficult in entrance examination and very, very tough. Uh, uh, Mainly because it's very tough and expensive, because you have to send your child to the city where the medical school is located.
Mm.
And that's in the mainland of Korea,
Mm.
where living cost can be very expensive compared to life in Jeju.
Mm.
So I was discouraged to apply uh, in medicine and medical college.
Mm.
Yeah. So
So, but
that
you did go to Seoul National and you got a
yeah,
bachelor's and master's. So tell us how that came about.
it came about. I was thinking about alternative area to to pursue, and in Jeju City at that time there were many uh, people and industries uh, moved from Seoul to Jeju Island because of the uh, Korean War,
Mm.
and we had a house in two parts, and one part was for rental, and a family moved into that rental section of our house. And turned out that the man was the president of a company producing uh, soybeans paste,
Oh,
soybean paste.
yeah, yeah,
Yeah.
yeah.
So soybean producing company
Mm.
very close to our house.
Mm.
So he was a company president.
Mm.
So that's how it came about.
And so how long did it then take to get a bachelor's and master's?
Bachelor's in 1974.
Mm.
Bachelor's for after 4 years.
Mm.
Yeah. Yeah, I went through 4 year course, master's. Um, right after bachelor's 4 years, I I joined the Korea Military Academy
Oh,
Institute
okay.
of Research, Korea Institute of Research for Defense Department.
Mm.
And that meant that I had to be a private soldier in the army. So they allowed me to pursue my master's program while serving on the military duty.
Wow.
So when I finished the 2 years of military duty, I also finished my master's degree.
I see.
So I'm thankful to the director of the institute allowing me to do this. Yeah.
Wow, yeah. What was
So
your research about?
the biosynthesis of flavins
Oh.
in
Oh, okay.
soybean sprouts.
Okay.
So that was my research project.
Yeah, so that's how you started your interest in flavins.
Yeah. Yeah.
So then you came to the US and Davis. And how did you choose Davis?
I applied to the UC Berkeley,
Oh,
and they accepted my entrance, but to the Davis campus.
oh.
They looked at my bachelor's background,
Mhm.
and apparently the graduate recruiting committee decided that my interest would match the Davis campus better than Berkeley.
The agricultural angle. Yeah.
Yeah, and so that I ended up in Davis, California.
Mhm. What work did you do there?
Entirely different research as a graduate student. I worked on what's called the Maillard reaction.
Oh, yeah. Yeah.
Maillard reaction, you know,
Yeah.
the browning reaction.
Yeah.
When you bake your bread, it browns.
Yeah.
The chemical mechanism of that.
Yeah.
Extremely complicated chemistry.
Yeah.
So while I was doing that research, especially at the early part of the research, I decided I was not going to do anything significant in this field. So I kind of directed my research on the inhibition kinetics of the Maillard reaction. So I did mostly kinetic experiments like heating glucose solution to its browning reaction. Yeah.
Yeah. No, real fun. How did you analyze the products?
Well, the analysis was not very successful because it's too many different products.
Oh, I see.
So that's why I focused on kinetics simply by following colorimetrically the absorbance increase in the visual range.
Sure. You don't have to know what's going on, just that it's going. Yeah.
Yeah.
So, but thinking about the English language, where did you learn how to speak English?
Yeah, that was very tough at first because these days most of the Korean graduate students or undergraduate students wanting to study in the United States, they speak very fluent English.
Yeah.
But when I was a graduating senior, English was so difficult. One of the English conversation or English speaking tested was interview session with the associate counselor of the American Embassy.
Wow.
In Seoul. He was invited as the examiner for the invitation because I was one of the 21 graduating students selected by the government to be sent to the United States. So this counselor, associate counselor, asked me a few questions in English. I was very, very lucky that I understood his questions. So for example, I remember this how do you get to Busan?
Oh.
Busan is the largest city in the south part of Korean Peninsula. And I understood this, even though I didn't know the English, I understood it. So I said, by train, by train. And so he followed this question with similar kind of questions, and I answered in one word, one phrase, by train. Yeah.
Okay, so you've got then now a PhD. How did you choose your postdoctoral mentor, and what was it like in Ames? And what was David Metzler like?
David Metzler. I chose Clinton Chichester as faculty advisor.
Ah,
Yeah.
nice.
And uh, and the main reason for that was that there was an intercampus, uh, uh, there was a campus faculty group, program group called agricultural biochemistry,
Mm,
and he was the head of that program.
mm,
So uh, that was the reason why I chose him as
mm,
my faculty graduate advisor. Yeah.
mm, mm.
Uh, and what's the other part of the question?
So the postdoc,
Postdoc, yeah, David Metzler.
yeah.
For two
So
reasons I chose him.
yeah,
One was that he was, and probably still is, the author of the book Biochemistry,
yeah,
textbook of biochemistry.
yeah.
It was the most well-adopted book in the U.S. Universities at the time.
Yeah.
So he was a very famous name for me.
Mm,
And secondly, he had an ad seeking postdoctoral fellowship candidate, and
mm,
he mentioned in that advertising that he was interested in the um, photochemical reaction
oh,
of flavins
oh.
in in solution,
Mm,
not in food or anything, but in solution. So
mhm.
I applied for these two reasons. One, he was very famous biochemist,
Yeah.
uh, and secondly, he was interested in flavin chemistry.
Well, when I was an undergraduate, we had his textbook in my biochemistry course.
Okay, yeah, yeah, very well known book at the time. Yeah,
Very well known.
yeah.
Let's see. How about your first paper? I did not look it up, but I hope you remember your first paper. When did you write your first journal article?
It was when I was a research member of the Korea Military
Oh, okay.
Institute, Scientific Research Institute,
Mhm.
and it was about the biosynthesis of flavins,
Mhm.
and published in the internal journal.
Oh,
Uh, name of the journal was the Journal of the Institute of Scientific Research.
mhm.
The publication was sort of like institute's own journal,
Mhm.
but in Korea there were very few scientific journals. In fact, almost none.
Mhm.
So this journal became very well-known journal outside of the institute.
Mhm.
So officially, that's my first publication in my bibliography. Yeah.
Great. And so you wrote the whole thing, or was there another author?
Oh, with my there was a supervisor,
Yeah.
uh, 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 Dr. Dr. Choi,
Mhm.
Choy.
Mhm.
Yeah. So he and I wrote the paper. Yeah.
So then, thinking back, how far did your research as a professor diverge from your graduate and postdoctoral work?
Immediately after graduation, because I didn't like the field of Maillard reaction. So I escaped it to go to Ames, Iowa.
Yeah, yeah.
Yeah.
So yeah, good choice. So we have then the final section about the personal things.
Yeah.
Tell us about your family. Did you marry or have a partner? I know you did. And then
Yeah.
children.
I have a wife. Uh, I have a daughter living in Miami, and
Okay.
as a ophthalmologist,
Oh, really?
and I have a son living in Mülheim, Munich, Germany.
Oh,
I rarely see him because of the distance
yeah.
and difficulty of long travel for my 90-year-old
Yeah,
body.
right. So where were they both born? I mean, you've moved around a lot. Where was it both in Texas?
Both of them were born in Texas. Yeah.
Okay.
Uh,
And
no, no,
then
no. I'm
no?
sorry.
Yeah.
My daughter was born in Lincoln, Nebraska.
Oh, okay.
Uh,
Yeah, I know she was little when I met her.
yeah,
That was a long time ago.
yeah.
Before retirement, what did you do for relaxation or entertainment?
For relaxation, I play golf whenever I can,
Mm,
assuming that I can assemble my golfing partners
mm,
and uh, and I guess I read a lot. I read enjoying books,
mm,
both in English or in Korean.
mm.
Many books to read these days,
Yeah.
and I spend a lot of time reading
Mm.
this book.
So since you retired then, you're still doing the same things that you've always done to relax.
That's right.
Yeah.
I also swim every day.
Oh,
Not every day, but I at least three, 4 times a week.
oh, wow.
I swim every day.
Mhm.
I swim at the nearby swimming pool.
Mm,
But I enjoy golf
mhm,
whenever I can. Yeah.
mhm. All right. Well, that is the end of the interview, all my questions. And thank you
Yeah.
very much for talking to me today.
Thanks a lot. My memory is still serving me good. I think I can remember your voice. So maybe I don't have to worry about Alzheimer's.
Yeah. No, you're doing pretty good. You made it pretty far.
Uh,
All right. I'm
okay.
going to